Hello, my name is Colleen Amundsen,
Hi, I'm Shannon Bachman.
and we're going to be discussing the autonomic nervous system. So, the autonomic nervous system is made up of the sympathetic, the parasympathetic, and enteric nervous systems. This system as a whole controls the visceral functions of the body. This includes arterial blood pressure, GI motility, bladder control, sweating, body temperature, and others. It has the ability to change these visceral functions in a matter of seconds. It controls these functions from centers in the spinal cord, brainstem, and hypothalamus. The third part of the autonomic nervous system is the enteric nervous system. I just wanted to say a little bit about that, although we won't be focusing on that part of the system. The enteric nervous system controls the gut, spleen, pancreas, and liver. The enteric nervous system's blood flow is supplied by the splanchnic circulation. The splanchnic circulation is innervated by neurons originating from the celiac, superior mesenteric, and inferior mesenteric ganglion.
Now, as we said earlier, the autonomic nervous system has two subdivisions, as you can see here, the sympathetic division and the parasympathetic division. The sympathetic nervous system will be discussed by me, and in this video, we hope to give you a thorough explanation of each division and its respective effects on the body. The sympathetic nervous system is known as your flight or fight response. This is what kicks in when you need to have a lot of energy and get your body moving quickly. It originates in the spinal cord between T1 and L2, which you can see here. This will be T1 and down here will be L2. It can also be called the thoracolumbar tract. It has direct innervations with the eye, the salivary glands, the heart, the lungs, blood vessels, liver, stomach, pancreas, adrenal medulla, the bowel, bladder, rectum, and reproductive organs. Specialized sympathetic nervous system fibers also innervate the piloerector muscles and the sweat glands. It can be appreciated from this image that T1 innervates the head directly. You have T2, which innervates the neck. T3, 4, 5, and 6 are the thorax. T7, 8, 9, 10, and 11 innervate the abdomen, and T12 through L2 innervate the legs and the lower portions of the GI tract along with the reproductive organs. Now Colleen will explain the parasympathetic.
Okay, so the parasympathetic nervous system is your rest and digest system. It works in conjunction with the sympathetic nervous system to maintain homeostasis within the body. When you think of the parasympathetic nervous system, some of the effects that it has on the body includes a slowed rate and decreased force of contraction of the heart and dilation of the coronary arteries, constricted airways, and this is because the body is not in the fight or flight mode, so it doesn't need to dilate the airways to increase oxygen exchange, increased sweating, increased secretion of enzymes in your glands, and pupillary constriction. It has little to no effect on basal metabolism, abdominal viscera, blood vessels, mental activity, and skeletal muscles. The areas or levels of the spinal cord associated with parasympathetic outflow are the cranial region and the sacral region. And we'll go to the next picture to talk a little bit more about this. Okay, so this picture just shows a little bit more in detail the parasympathetic system. The cranial region does the majority of the work for the parasympathetic system. It involves cranial nerve 3, which is your ocular motor nerve, and this provides stimulation to the pupillary and ciliary muscles of the eye. Also, cranial nerve 7, which is your facial nerve, provides stimulation to the lacrimal, nasal, and submandibular glands. Cranial nerve 9, which is your glossopharyngeal nerve, provides innervation to the parotid gland. And finally, cranial nerve 10, or the vagus nerve, which houses 75% of all parasympathetic fibers and innervates the entire thoracic and abdominal regions of the body. This is your major nerve of the parasympathetic nervous system. The sacral region fibers are pelvic nerves, which innervate the descending colon, the rectum, urinary bladder, and portions of the uterus. It also innervates the external genitalia. One of the most important effects of the parasympathetic nervous system is the ability for a strong parasympathetic stimulation to dramatically reduce the heart rate to almost zero. In some instances, especially in the pediatric population that is extremely parasympathetically driven, it can stop the heart completely or for a few seconds and cause a temporary loss of all or most arterial pressure. One example of this is the ocular cardiac reflex. When there's traction put on the eye, the vagus nerve sends a signal, this is your afferent fiber, to the brain, causing a decrease in heart rate. Another effect from a strong parasympathetic stimulation is when there's traction put on the gut from the surgeon during a procedure. This traction also sends an afferent signal to the brain via the vagus nerve again to cause a vagal reaction of the heart, such as a decrease in heart rate. These examples demonstrate how much innervation the vagus nerve has within different organs of the body.
Here you can see the sympathetic nervous system with respect to its location to the spinal cord. Now, the sympathetic nervous system preganglionic neuron has several routes of travel. Of great importance is the location of the cell body. As you can see here, the cell body is located in the intermediolateral horn of the cord, and that would be right here on the image here. And that'll be important as you're discussing the innervation of the sympathetic nervous system in relation to the cord, the brain, and the body. And we'll get through all these further discussions of its innervation with the postganglionic neuron as we move on. But you can see here, just labeled for you, is the ventral root, the white rami, the gray rami, and their different synapses along with routes of travel. Now here, the sympathetic nervous system, you can see that the cell body is located in the intermediolateral horn of the cord, and that's here highlighted for you in black. Its fibers pass through the anterior root of the cord, following its corresponding nerve fiber. Along the spinal nerve, a corresponding spinal nerve. After the spinal nerve leaves the spinal canal, the preganglionic sympathetic nerve fiber passes through the white rami, and it's white because it's myelinated, and enters the sympathetic chain ganglion. It then can do one of three things. So on this image right here, let me just get my pen going, you can see we're now inside the sympathetic chain that runs bilaterally along the outside portion of the spinal canal itself. From the white rami, it then can do one of three things. It can synapse with a postganglionic neuron in the sympathetic chain at the same level at which it enters. It'll then exit that level and travel out the gray rami and to its corresponding organ. It can synapse, it'll travel up a level or down a level through the sympathetic chain and synapse with a postganglionic neuron there, and again affect its corresponding organ. And you can see here exiting through the gray ramus, or traveling down a level, exiting through the gray ramus again, and going to its respective organ. The final option that the spinal nerve has of three is it can travel directly through, the preganglionic sympathetic nerve, is it can travel directly through the sympathetic chain and synapse with the postganglionic neuron in the peripheral sympathetic ganglion, as you see here. And this is an image from your coloring book. If you got it, I encourage you to use it. It definitely helps break this down for you. But here in 4, you can see it synapsing with the postganglionic neuron in the peripheral ganglion. Right there. Okay, so the postganglionic sympathetic nervous system fiber originates in the chain ganglion or in one of the peripheral sympathetic ganglion, as you can see here. The postganglionic neuron is considered to be the long neuron of the sympathetic nervous system, and the preganglionic neuron is the short neuron of the sympathetic nervous system, and that's a good way to differentiate sympathetic from parasympathetic. The postganglionic sympathetic neuron then travels to its respective organ, and of note, some sympathetic preganglionic fibers do travel back from the sympathetic chain into spinal nerves through the gray rami at all levels of the cord. So what that's saying is that it'll then follow a spinal nerve out to a skeletal muscle or a blood vessel, and these will control blood vessels, sweat glands, and piloerector muscles of the hairs, and they are cholinergic, as you'll be explained later. So here you can see, again, we're just talking about pointing out the different sympathetic chain ganglion, as you can see here, running bilaterally along the side of the cord. And you can see the celiac ganglion here, the superior mesenteric ganglion here, and the inferior mesenteric ganglion here. Innervation of these ganglion are directly linked to the the liver, the pancreas, stomach, bowel, and bladder, the adrenal and kidney glands, and reproductive organs. It's important to note that when you affect something through this chain ganglion, it doesn't affect only one particular organ of the body. It will affect a general amount of organs in the body, meaning the kidneys and the adrenal gland together, or the bowel and the bladder together, things along that idea. Now you can see here again, another important thing to point out with the sympathetic innervation is the cardiac innervation. This is important to note that the cardiac accelerator nerves are T1 through T4, and that will be important when you're considering neuraxial blockades. You don't want them to go above T4, or you're wiping out your cardiac accelerator nerves, leaving only parasympathetic to activate this, and can actually lead to bradycardia or an asystole. And you can see them there. So here, again, this is just a brief explanation of the sympathetic and parasympathetic, how the fibers are related, what neurotransmitters you're looking at, and what receptors you're looking at. With the sympathetic, you can see here that we first have a short preganglionic nerve secreting the neurotransmitter acetylcholine, activating a nicotinic acetylcholine receptor on the postganglionic sympathetic fiber, which then releases norepinephrine into the effector organ. Those organs will have either alpha or beta receptors and to create their specific response. This is known as the adrenergic system because of the norepinephrine here. Now, just briefly, we'll go through alpha and beta, and then Colleen will explain what the GQ and GS coupled means. But I just want you to know that alpha has some subdivisions associated with it. You have alpha 1 and alpha 2. So we have 1 and 2 here, and this is GQ coupled, and then alpha 2 is GI coupled. And like I said, Colleen will go through and explain them to you. When alpha 1 is stimulated, it affects blood vessels. It causes vasoconstriction. It can also cause iris dilatation, GI and relaxation, GI and GU sphincter contraction, and coronary artery constriction. Alpha 2 is a modulating receptor, and that actually will inhibit further neurotransmitter release. Now with beta, you have beta 1, beta 2, and beta 3. Beta 1 and beta 2 are the ones that we most primarily affect in our anesthetic. Beta 1 is GS coupled, and beta 2 is GS coupled. Stimulation of beta 1 increases heart rate and contractility, and stimulation of beta 2 will cause vasodilatation, GI relaxation. Most important is the bronchodilation effects of it, uterine relaxation, glyconeogenesis, bladder wall relaxation, and coronary artery dilatation. Beta 3's primary role is in thermogenesis. It's important to note that norepi and epi affect the beta receptor equally, but the alpha receptor is most notably affected by norepinephrine, less by epinephrine.
Okay, so Shannon just described the sympathetic system, or the adrenergic system, and conversely, the parasympathetic system is also called the cholinergic system. The primary neurotransmitter for this system is acetylcholine, seen here as ACH. Unlike the more complex sympathetic nervous system, the parasympathetic system has a very long preganglionic neurons, that travel all the way from the spinal cord to the end organ, whether it be the heart, lungs, eyes, etc. This differs from the sympathetic system, having to go through the different sympathetic chain ganglions, etc. The receptor at this junction is the nicotinic acetylcholine receptor. This is an ionotropic receptor, which means that it has a channel allowing sodium ions, in this case, to enter the cell and cause depolarization of the postganglionic neuron. The postganglionic neurons of the parasympathetic system are very short. They are actually in the walls themselves of the end organs that they innervate. The receptor at this junction is the muscarinic acetylcholine receptor, which is a metabotropic receptor, which means that it works with the G protein second messenger system to create changes at the cellular level instead of the direct ion channel effect of the nicotinic receptor. There are 5 different muscarinic receptors, M1 through M5, and this slide shows it a little bit better. M2 is a GI receptor causing inhibitory changes specifically in the heart. Anticholinergic agents, such as glycopyrrolate or atropine, specifically work at this receptor to block the cholinergic effects of some medications, such as neostigmine, and I will talk about a little bit more about that in just a minute. As a review, acetylcholine is synthesized in the terminal endings and varicosities of the cholinergic nerve fibers, where it is stored in vesicles. The chemical reaction that creates acetylcholine is acetyl-CoA plus choline with choline acetyltransferase. After the acetylcholine is secreted into the cleft, it is broken down by acetylcholinesterase into an acetate ion and choline. And as always, the action potential spreads over the terminal fibers, depolarizing them, which increases the permeability of calcium ions into the cell, which causes the secretion of the neurotransmitter, in this case acetylcholine. So we always think calcium in, neurotransmitter out. So as Shannon was discussing earlier, the alpha and beta receptors were G protein receptors or metabotropic receptors. I just wanted to talk a little bit more about what that means. So there's 3 different types of metabotropic G protein receptors: GS, GI, and GQ. GS is stimulatory, S for stimulatory, and its actions are produced by a stimulation of adenylyl cyclase, increased CAMP, increased ATP, which this whole process causes intracellular changes to occur. GI, or inhibitory, is just the opposite. It's just what it sounds. It's an inhibitory protein. So this does the exact opposite. It inhibits adenylyl cyclase, inhibits CAMP, and it inhibits ATP, preventing intracellular changes. Sorry about that. And finally, there's GQ, which works in a little different manner. It is stimulatory as well, and although unlike the other 2, it's activated by phospholipase 3, IP3, and DAG pathways. The muscarinic receptors that are located at the postganglionic neurons of the parasympathetic system are, as I said, M1, 2, 3, 4, and 5. M1, 3, and 5 depolarize and are GQ coupled. M2 and 4 hyperpolarize or inhibit, and therefore are GI. And we think of our M2 receptor as one of the main muscarinic receptors, and this is mainly in the heart, and it's affected by the vagus nerve.
So here you can see a little bit more of a breakdown regarding the autonomic nervous system, sympathetic and parasympathetic, along with its corresponding receptors. This is just supposed to be another visual for you to look at in case the last ones didn't catch your eye. I did want to go through a little bit of the synthesis of norepinephrine and epinephrine, just to give you background on that. So norepinephrine and epinephrine, the synthesis of, begins in the axoplasm of the terminal nerve endings of adrenergic nerve fibers. It is completed inside the secretory vesicles. So you need tyrosine, which is a non-essential amino acid, and that's converted to dopa via hydroxylation. Dopa is then converted to dopamine via decarboxylation, and this dopamine is transported into vesicles where norepinephrine is formed by the process of hydroxylation. Now in the adrenal medulla, 80% of norepinephrine is converted into epi via the process of methylation. And after secretion, it's important to know that norepinephrine is removed from a site in 3 ways. The first way, and the most important, being reuptake into adrenergic nerve endings by active transport, and this is about 50-80% of your norepinephrine is removed in this way. It can also be removed from the site of action via diffusion or by destruction of tissue enzymes, monoamine oxidase, which is located in your nerve endings, and catecholomethyltransferase, which is present diffusely in all tissues. So again, this is just another look at your different receptors. So that's the end of our autonomic nervous system lecture. We'll be happy to mail this out to you all. And please, if you found something that may not be what you were taught in class, let us know and we can clarify anything we said. Thank you very much for your time and good luck studying.